Table 10.6 of your textbook summarizes the classification of different functions of proteins as well as the description of those functions and examples of each. So these functions include messenger proteins, receptor proteins, transport proteins, storage proteins, contraction proteins, protection proteins, structure proteins, and catalysis proteins. So a hormone is a chemical that is sometimes a peptide or a protein that is created in one part of the body that affects another part of the body. An example of a hormone would be insulin, and that is a protein. It's released from the pancreas and it goes to other cells throughout the body. Receptors are proteins that face the outer surface of a cell and bind to a hormone or other messenger chemical, triggering a signal inside the cell. So there are insulin receptors that are present on the outside of a cell, and they will bind to insulin that was sent through by the pancreas. And when they bind to insulin, it triggers the uptake of glucose into the cell. And the glucose transporter is a protein that is an integral membrane protein spanning the phospholipid bilayer of a cell, which transports glucose from outside the cell into the cell. So we can see that insulin is a messenger and it binds to a receptor and then triggers the work of a transporter protein. Another type of transporter protein is hemoglobin, and this is the protein that transports your body's oxygen throughout the body. Hemoglobin is made of 4 polypeptide subunits, and they are signified here by the blue and the red. And these curly ribbons represent the secondary structure of each of those individual polypeptide subunits, and they're held together through the quaternary structure to make up the structure of hemoglobin. That quaternary structure is held together by hydrogen bonds, by salt bridges, as well as London forces. Also in the structure are 4 heme groups, which are shown by these green structures. The heme group is not a protein, and it's known as a prosthetic group, but it is an important part of the structure. Each heme group binds iron plus 2, and then that iron plus 2 binds to an oxygen molecule. Each iron cation can bond to 1 oxygen molecule. When the 4 iron cations are bonded to oxygen, hemoglobin undergoes a conformational change, meaning its structure changes. When the hemoglobin reaches areas of the tissue that are low in oxygen, the oxygen leaves or dissociates from the heme group, and then the conformation of the hemoglobin changes back to its deoxygenated state, and it's ready to bind to more oxygen molecules.